Thanks for the opportunity to talk about some of the new exciting MSA research. Multiple system atrophy affects multiple types of brain cells in more than one region of the brain. And the two main brain cells include the supporting oligodendroglial cells and the neurons that communicate all your thoughts and actions. And these micrographs on the right show the small oligodendroglia with inclusions, and then on the left, the neurons with some of these supporting oligodendroglia inclusions around them. The oligodendroglia make abnormal aggregates of the alpha-synuclein protein in inclusions, which are shown in the micrographs. And the neurons have increased alpha-synuclein protein, not necessarily always in inclusions, which are shown in the neurons. So I was going to talk today about 5 different aspects of new research. The first things are how are the cells affected in MSA? And this type of research allows us to identify the problems, and therefore we can target them for potential treatment. What are the cells that are most effective? And this allows us to focus on measuring those particular focal areas for more effective treatments. There's some new animal models that are good tools for MSA, and these can allow us to now look at neuroprotective therapies using these new tools. And importantly, the diagnosis of MSA needs to be earlier, and this will allow us to do therapeutics at a time when it will make the most difference to people. And then lastly, what's new in therapeutics for MSA? So the first thing to think about is the problem is the protein, it's alpha-synuclein. And alpha-synuclein is the important protein that's involved in regulating the chemical connections between the two neurons, between all your neurons. The protein abnormally aggregates, but it doesn't do this just in multiple system atrophy, but it also occurs in people with Parkinson's disease as well as in people with dementia. So a problem with the protein itself doesn't give you just multiple system atrophy. Now, the difference between multiple system atrophy and these other diseases is in how the protein is structurally abnormal. And again, the photo of the oligodendrocytes that are abnormal on the right versus a Lewy body inside a cell in Parkinson's disease. And you can see the structures look quite different. And this is actually even different down to the very minute fibril type of structure that also are different between these different inclusions. So not only are the structures unique to the disease, but in multiple system atrophy, there's more than one of these different types of structures, at least 2, and perhaps more. And so we know that these different structures, and particularly the 2 different types of structures that we've identified in multiple system atrophy, increases the aggregation and makes it more toxic. Another thing that's been found is that there's a particular proteins in the oligodendroglia, and one of them, the P25 alpha, actually mixes with the alpha-synuclein, and that also makes the inclusions different and also highly pro-degenerative. And this is just a cartoon of the P25 alpha in how they interact to form the inclusions. Knowing this, we can now target this particular unique aggregation of the protein. And then some of the proteins in oligodendroglia have also been noticed to affect the changes at the DNA level. And the DNA codes for the protein so that it changes how the amount of protein that's made. And so this can also now be targeted for novel therapies. And lastly, the increased alpha-synuclein that's found in neurons has been shown to be cut off at one of its ends, giving it a unique property and also the potential to initiate aggregates. And this form of alpha-synuclein can also now be targeted. So where are the cells that are most affected in the brain? The cells that are most affected are the oligodendroglea that are undergoing the maturation. So they are like teenage oligodendroglea rather than mature. And they're specifically impaired in MSA. And it leads to insufficient replacement of more mature oligodendroglea, which the brain relies on to support the neurons. These immature oligodendroglea are actually very prone to take up the aggregated alpha-synuclein. They also have an impaired capacity to release small vesicles known as exosomes. And the exosomes have been known to be a way that the cell can get rid of abnormal protein. And so by having a problem with this capacity, it would increase the cellular alpha-synuclein in the oligodendroglea. And in regions of the brain where the oligodendroglea accumulate alpha-synuclein, there are increased numbers of the antigen-presenting microglia, the immune cells of the brain, and also invading peripheral immune cells. This suggests that the oligodendroglea becoming abnormal actually generate an immune response in the brain. And there's new imaging techniques that are able to detect these degenerative changes in oligodendroglea in the white matter, and that correlates with the severity of clinical symptoms. These are now tools that can be used to track the disease severity. And the changes in the white matter correlate with the decreased level in serum urate in the blood. And this suggests that maybe urate, it could be a potential neuroprotective factor for white matter degeneration in multiple system atrophy. As I said, there's new viral-based rodent and non-human primate models of multiple system atrophy that have been developed, and they reproduce the pathology of multiple system atrophy with high fidelity. And these models use a new novel virus, and they're constructed to infect the oligodendroglea in the regions most affected, most impacted, and they increase the amount of alpha-synuclein the oligodendroglea make. And so these new models are now being used for testing novel therapeutics aimed at reducing this pathology of MSA. And importantly, trying to get diagnosis right for MSA is critical. And there's been a number of new developments, the assessment of the type of the alpha-synuclein aggregate and the levels of an important neuronal protein, neurofilament light chain, in cerebrospinal fluid, which is the fluid that bathes the brain. And this can differentiate early MSA from controls and patients with the Lewy body diseases, Parkinson's and dementia. The neurofilament light is significantly higher in MSA, and alpha-synuclein has the different properties in MSA, which we discussed earlier, the earlier aggregation and difference in protein conformation. And there's a new blood test that measures the alpha-synuclein in the blood exosomes, those small vesicles, as identified coming from either neurons or oligodendroglea, and these can distinguish patients with MSA from those with Parkinson's disease. And lastly, assessing all of the blood cells shows that there's an early reduction in one of the type of white blood cell, the intermediate monocytes, and that the changes you can also measure in the blood changes in antigen presentation from the brain. And this is indicative of that active neuroimmune response, which then can be monitored for therapeutics. So these developments give us a way of looking at blood to see if we can modify them with therapeutics and the representative of brain imaging or blood tests. So, as it said, in addition to developing tests like imaging and blood tests, diagnosing MSA at the first onset of symptoms is problematic. And new research has identified that imaging the brainstem or cerebellum and the presence of new or increased snoring is highly predictive of MSA at an early stage when it presents to the clinic. In terms of therapies, there's been new therapies that are looking at helping the symptoms of MSA. These have been small trials of rehabilitation. They appear effective for alleviating some of the symptoms. A 5-day inpatient physiotherapy program followed by a 5-week unsupervised home-based exercise program in 10 patients showed improvements in gait performance. And in-hospital physical rehabilitation using physical, occupational and speech therapy in 15 patients showed significant improvement of ataxia or balance, of cognition and of health-related quality of life. So these rehabilitation strategies look like they are improving some of the symptoms. Unfortunately, there's no improvement in emotional health for the patients. And I guess disease-modifying therapies may be more effective at that. And lastly, there are some interesting new neuroprotective therapies that are in clinical trials. There's 2 new active immunotherapies that are vaccinated against the alpha-synuclein protein. They've been trialled in a 52-week phase 1 study of 30 patients, and they were found to be safe and well tolerated. And one of the vaccines produced a rapid and long-lasting antibody response that specifically targeted alpha-synuclein. And so this shows great promise, and hopefully more trials like this will occur. In addition, low vitamin B12 levels have been associated with shorter survival, and these may be amenable to modification. And the use of non-aspirin, non-steroidal anti-inflammatory medications is associated with reduced risk of multiple system atrophy. So there's a few things that are actually coming up that are actually look like they could be helpful in neuroprotective strategies for multiple system atrophy. And thanks very much for listening.